Hello, my name is Phil Brannen, and I'm a plant pathologist with the University of Georgia. And I'm going to be talking to you about sampling for botrytis and anthracnose resistance. And so there are several things you need to know about this that are be important as far as making this successful for you. And so we'll go through this slide set, and hopefully by the end of it, you'll have all the information you need. Guido Schnabel with Clemson University and established the initial resistance profiling programming that we have been utilizing for some period of time in the Southeast. And this was mainly designed for botrytis, and so it's been utilized very effectively. The reason this was done, if you look at the why we would do this, is first of all, location-specific resistance profiles make growers aware of potential problems. This allows you to customize your recommendations. It also utilized educational experience from experts to help implement the IPM program in your particular strawberry sites. And implementation of resistance management helps keep fungicides efficacious. That helps to keep them on the market for longer. And also, any of these monitoring results you receive from the specialist will help you to assess your impact and outcome from the recommendations you receive. So it's a good opportunity for us to interact with U.S. producers to help you implement integrated pest management and resistance management in your programs. More recently, the Plant Molecular Diagnostics Lab down in Tifton, Georgia, has taken over this process from Clemson, and MD Ali is the leader of that group. And this lab is now capable of running the same basic analyses on botrytis, and also I'll talk to you about anthracnose and what they're doing for that as well. But you can send samples there from throughout the Southeast, and these samples will be analyzed. As of right now, there's actually funding that is provided by the Southern Region Small Fruit Consortium to help pay for some of these samples. Once that money does run out, though, the samples are on a fee basis, and so it would be a good idea for you to get your samples in early to make sure you're covered by the funds that are provided by the Southern Region Small Fruit Consortium. As far as samples you would take from strawberry for botrytis, these are probably the easiest ones to take as far as plant samples. So these blossoms or leaf samples where you can see the fungus sporulating, that would allow you to send these in directly, and you could do that. There's also the possibility of sending fruit. However, fruit is not a good idea because actually what's going to happen is all this fungal growth is going to go into the fruit and basically just turn it to mush. So there's another way to handle the botrytis that you're getting off of fruit. So I'll talk to you about that. But as far as the samples you can send, there's really two or three ways you can send these samples. One is to send these dead strawberry flowers from the plants where you see these with the botrytis growing in the flower. Also, you can find this sometimes on leaves as well. But you want to send enough that the lab will be able to fully isolate from the botrytis organism and make sure they get a good collection of materials from that. The other way you can do this is to send swabs, and I'll show you another example of this in a minute. But you can take Q-tips or basically any kind of swab and just take a very light rub on a strawberry where the botrytis is. You don't want to dig it into the plant or into the actual fruit because that's just going to be a mess. So you're just rubbing off spores lightly onto the swab itself, and that's what you would send. Now, I personally would like to see each of those swabs independently sealed in a Ziploc bag, a little small Ziploc bag, such as a sandwich bag or something like that, where you can seal them. I prefer to keep them separated because that way you're actually getting separate swabs from throughout the field. And you'd like to kind of go throughout the field in a Z-type pattern throughout your strawberry patch and randomly assess or gather those swabs and then put them in individual bags. By the same token, I think it's better to go, if you're going to use dead leaves or flowers, do the same thing. I'd prefer to see that you isolate this into 10 different segments that you would send in so that the at least 10, so that the lab can get at least 10 isolates so they can then use for their analysis. So that's how I would like to see that done. If you put them all in one bag, the issue there is you might be mixing spores, and so you might get a false understanding of what's going on throughout your entire area. So that's what I would prefer to see you do. As far as collecting in these swabs, if you have fruit like this with the botrytis on it, you can just take these swabs and again rub that lightly onto the fruit get spores, but you don't want to dig it into that rotting fruit because that's just going to be a mess. And it may be much more difficult to isolate. And then you're going to get all kind of other secondary growth on these swabs as well. So you just want to lightly rub it, get enough spores that MD Ali and his group down there can actually grow this out. But that's what you want to do. You can also do this for blackberries. I know this is a strawberry meeting, but you can do this for blackberries in a similar fashion just by sending in swabs. Same thing for grapes as well. All of these different plants that are small fruits can get botrytis. And again, for right now, those are covered by the funding that has been provided by the Small Fruit Consortium until the funds run out. Also have botrytis on blueberries as well. And it's good to know in all these commodities what your resistance profile is so that you're not spraying fungicides that have no activity on this pathogen whatsoever, and you're just wasting money and effort, and you're still going to have a lot of disease at the end of the day. As far as what's happening when it gets to the lab, this is what the resistance profiling system kind of looks like. So there'll be a different grouping of fungicides across the top, all different kinds of activities for these fungicides, different classes of fungicides. And then you would have a standard control where no fungicide is applied at all in the well. So this would be an agar media. And then this is an example here showing where the control would definitely grow out. And then here, some of these fungicides would have activity and probably some would not. When you go to the next slide here, you kind of see what that looks like. Again, here's your control. Here's different fungicides. You can see the different levels of disease that are afforded. Some of these are not active at all. So, for example, here, the thiophanate methyl, which will be found in Topsin. Here you see perfectly good growth here that is comparable to a non-amended media. But then you look at some of your other materials over here, some of the SDHI type fungicides. Some of these are very, very active. So you can kind of get an idea of what's working and what's not working. And again, that helps you to make sure you're spraying the right fungicides for botrytis. Another fungus which we're also able to test for at the lab as far as resistance development is Anthracnose. We do know in particular that the quinone outside inhibitor fungicides, the QOIs such as Abound and Pristine, those particular products are often going to have resistance to them with the anthracnose fungus, the Calototricum type fungi that are found on strawberries. So this is something else we can also do through the lab in Tifton. For here, in this case, you are going to be sending the fruits, okay? And so hopefully they will not rot like botrytis. This is a kind of a hard rot as opposed to a soft rot. And so if you do send fruit, what I would suggest you do is make sure you individually bag those fruits, just as I told you in the beginning. Make sure you put an individual strawberry in an individual bag. You would want to go throughout your strawberry planting in a Z-type pattern across the area to take these samples. You might want to send in a few more than 10. They're probably only going to use 10, but 10 to 20 symptomatic strawberry fruit would be good. That way you make sure they get good samples for them to look at. And then also, I would like to see you send these overnight just to make sure that they do not rot. That's going to be an added expense on your end, but it will more than likely allow them to get these individual fruit out without them rotting in transit. And so that's why I would say to do that. Try to use overnight delivery as opposed to sending them over a 2 or 3 day period, which they might actually rot, and then you wouldn't get anything out of it. You can also sample anthracnose on blueberry, and we're seeing resistance to QIs there as well, and also in wine grapes. This is another example shown here of anthracnose, again, Calototricum species that are developing resistance and where we're having issues. We see this on Vitis vinifera grapes and also muscadines now where resistance is developing. It'd be good to have that kind of testing going on as well. This is all the information on shipping and mailing and phone numbers and everything for MD Ali and his lab. And so this should give you everything you need with the information I provided to think through how you would actually sample and also send samples to this lab for getting your resistance profiling done on strawberries and also on other commodities as well. So again, this completes my presentation. Thank you for your time. And if you have any questions, I'll be glad to answer those if I can. And at this time, though, we'll turn it over and take questions.